Please, we shall begin this event with a prayer. This prayer will be said individually and silently. May God in His infinite mercies accept our prayer. At this juncture, please kindly rise for the national anthem.
Nigeria, we hail thee, our own dear native land. Though tribe and tongue may differ, in brotherhood we stand. Nigerians home and proud to serve our sovereign motherland.
University of Abuja, for unity and scholarship, in pursuit of excellence, molding models of character, cultivating culture of integrity, known for blessing on human endeavors. In honor we center, holding the torch of erudition. As we give light in the paths of greatness, strength and virtues for all mankind. Our cherished great Uniabuja, Uniabuja, is fountain of knowledge for all. Fountain of knowledge for all. Fountain of knowledge for all.
Please be seated. The Vice Chancellor, members of the Council here present, principal officers of the university, distinguished guests, esteemed colleagues, professors, ladies and gentlemen, good morning. Well, it's my pleasure to welcome you all to the 8th Undergraduate Research Day of the University of Abuja, now Yakubu Gowon University. This important program was instituted to promote a strong culture of inquiry, innovation, and academic excellence among our undergraduate students. Today is a presentation of the findings of the undergraduate researchers who won the grants from the university at the 7th Undergraduate Researchers Day sometime last year. We recognize and honor our students who have distinguished themselves through dedication, intellectual curiosity, and the production of cutting-edge research in their various areas of specializations or discipline. Now, 6 findings will be presented today. But before we proceed, may I have the pleasure to introduce briefly to you the distinguished personalities who are here to witness this special occasion. Let me specially welcome and recognize the presence of an erudite and a consummate leader who is very passionate about the research development in our university. Ladies and gentlemen, please let's welcome Professor Hakeem Babatunde Fawene. Well, some of you have been meeting the Vice Chancellor, but I guess this clap is not. Please, let's step it up. More resounding clap for Vice Chancellor. Thank you, sir. Thank you so much. I like that. I can see the energy now. That's good. We also have sitting close to him a council member of our great university, a professor of environmental management, and the director of the Center for Undergraduate Research. Ladies and gentlemen, please let's welcome Professor Sunday P. Ejaro. Thank you. We have on the high table yet the acting director, Food and Industrial Biotechnology, Department of the National Biotechnology Research and Development Agency, Olukotun Debo Barnabas. Please let's welcome. On the high table also is the representative of the DG of NAFDAC from the Directorate of Planning, Research and Statistics Directorate, the person of Garba Usman. And I actually see here that he's not an ordinary Garba Usman, Chief Garba Usman. Please let's welcome him again. I can see that our acting registrar and secretary to council, Madam Sambo Mohammed, is here ably represented by one of our hardworking administrators, a deputy registrar in the university, Helen Ainda Yako. And another very distinguished personality here, who is the deputy director of the Center for Undergraduate Studies. Please let's appreciate Professor Perpetua Ibekwe. And by the way, she's one of our brand new professors. Please let's appreciate her more. Our great students, vibrant students who are gathered here for this special occasion today, please, I want you to appreciate yourselves very, very specially. Thank you so much. We shall go on with further recognitions of the presence of our great personalities as they come in. But in the meantime, may I have the honor to respectfully invite the director of this great center, Professor Sunday Peter Ejaro, to come forward for his welcome address.
Vice Chancellor, sir, the representative of the registrar, the representatives of the Director General of NAFDAC and National Biotech Research and Development Agency, other distinguished guests, members of the Center's Consultative Committee, staff of the university, our very cherished students, members of the media, ladies and gentlemen, good morning and welcome to the 8th edition of our Research Day. The mission of the Center for Undergraduate Research is to foster and promote a culture of research among undergraduate students, empowering them to develop solutions to challenges that contribute to national development. The theme of this Research Day which is promoting national development through research, aligns with this mission. The center also aims to translate students' research findings into entrepreneurial by collaborating with industry partners and our university center. As part of the center's mandate, annually we invite undergraduate students to bid for grants to undertake research in various thematic areas. In 2024-2025 academic session, that is last academic session, 6 teams were awarded research grants of various amounts after a competitive bidding process. Today, we are gathered to listen to them as they present their research findings. This event marks a significant milestone in the continuous growth and development of research culture within our university. Over the past year, these undergraduate students demonstrated creativity, ingenuity, dedication in their fields of study. Today, we are thrilled to showcase their research findings, which reflect the diverse talents, innovative minds that are shaping the future of our academic community and beyond. We appreciate our amiable Vice Chancellor for support the center. Thank you, sir. The research grant has played a pivotal role in nurturing the curiosity and ambition of these young scholars, providing them with the resources and support needed to explore new ideas, challenge existing paradigms, and contribute meaningfully to both academic knowledge and societal progress. The students you will hear today have not only invested their energy and time into their research, but they are also contributing to the ongoing discourse between academia and industry, bridging the gap between theoretical research and practical application. We are particularly delighted to welcome representatives from the industry today, represented especially by NAFDAC and biotechnology agencies. Your presence underscores the importance of collaboration between academia and the wider world. And we are grateful for the opportunities that such partnerships provide for our students. Your engagement with our research initiatives, whether through mentorship, funding, or feedback, is crucial to ensuring that the work we do here has tangible, real-world impact. We hope our guests will assist us in guiding the students on how their research findings can be converted to products. To the students presenting their research findings today, I want to congratulate each of you on the work you have done. You have shown resilience, curiosity, and a genuine passion for knowledge. As you share your research findings, I encourage you to take pride in your achievements and continue to push the boundaries of research and innovation. You are not just representing yourselves, you are also representing the excellence of our university and the promise of future progress. I would also like to extend my appreciation to the mentors and staff who have guided these students in their research. Your dedication and commitment to fostering a supportive research environment have been instrumental in their success. In closing, let us all take a moment to reflect on the significance of today's event. It is not just a celebration of individual efforts, but a reaffirmation of the power of research to drive change, solve pressing challenges, and inspire future generation of thinkers and innovators. The research findings being presented here today have the potential to shape the future. And I'm excited to see young scholars again to everyone for being here today. Let us continue to support and celebrate. Continue. Thank you once again to everyone for being here today. Let us continue to the transformative power of undergraduate research. I look forward to the presentations and the inspiring discussions that will follow. Vice Chancellor, sir, distinguished guests, colleagues, and students, I welcome you all. Thank you, and God bless us all.
Wow, thank you so much, Professor Ejaro, for that inspiring welcome address. Please, let's appreciate him once more. May I now invite the representative of the Director General and Chief Executive Officer of the Food and Industrial Biotechnology Department, Mr. Olukotun Debo Barnabas, for a goodwill message. Please, let's appreciate him as it comes to the ground.
Good morning, everyone. Vice Chancellor, the students, it's a delight. I'm glad to be here on behalf of my DG, Professor Abdullahi Mustafa, Director General, CEO of National Biotechnology Development Agency. He's very busy, couldn't come. But then looking at the importance of research, especially in the aspect of biological sciences, biotechnology, in national development. He has asked me to represent him, and on his behalf, he's wishing us the very best of the day. And I pray that as we go forward in research in Nigeria, just as the director said in his message, translating research into tangible product is the aim of national development goal. And so as students, as you key into the program of the faculty, you'll be taking the nation forward. And I wish you the best in all your undertaking. Thank you very much.
Well, thank you so much, sir, for your goodwill message. It's now my pleasure to invite to the podium our Vice Chancellor, Chief Executive Officer of our great university. Please let's welcome Professor Hakeem Babatunde for his address.
The Director of the Center for Undergraduate Research, Professor Sunday Ejaro, who is also a member of the Governing Council of this university. So when I call him my employer, he knows why. The Director, Professor Perpetua Ibekwe, the representative of the Registrar of the University, Mrs. Helen Ayinda Yakwo, the representative of the Director General and Chief Executive Officer of the National Biotechnology Research and Development Center, Professor Debo Olutokun, the representative of the Director General and CEO of NAFDAC, Chief Gaba Usman, very distinguished academics here present, our very dear students, gentlemen of the press, ladies and gentlemen, on behalf of the Governing Council, ably led by our Chairman of Council, Senate, Management, Staff, and Yakubu Gowon University, I want to welcome you all to the 8th Research Day holding here today. The theme of this gathering is very apt, promoting national development through research. Apt in the sense that research is one very important driver of industrial development all over the world in present times. And therefore, we know that and accept that today is a day of intellectual harvest. Today is a day of talent identification, where we are going to know some of our budding scientists and artists that we want to nurture to become world-class scientists in the near future. We are all aware that university stands on 3 tripods: teaching, research, and community service. And therefore, we take such occasions seriously, because it is very important to the survival of many nations. Research and innovative research, in fact, and creativity is what is driving industry throughout the world today. I therefore want to welcome you all while assuring you that today's efforts will improve the technological incubation and endowment portfolio of our university, and as you are all aware, will expand the frontiers of knowledge in the various disciplines for which these research findings have been made. It is on that note that I want to thank the director of the center, Professor Ijaro, for organizing this very important competitive research and for sustaining it, and the other facilitators in the center, which includes, of course, the team at the Center for Undergraduate Research. I thank you for inviting me to this occasion. As for me, it is a morning and afternoon that will be well spent. Thank you very much, and God bless you all.
Thank you so much, Vice Chancellor, for your address. Your address has actually strengthened the tone for this event. you have reaffirmed the university's commitment to research development in the university. Please, let's appreciate the Vice Chancellor more strongly. The next item on the program of event is a presentation of student, researchers, and this is very critical. There are about 6 of them, as I said earlier. We will be inviting each of the presenters to come forward. The first person I shall be inviting will be, has the research topic titled Application of Vermi Technology in Solid Waste Management and Sustainable Agriculture. This research has been led by a student from the Department of Microbiology. Ladies and gentlemen, let's welcome Yojo Nathan for the presentation. And he has been collaborating with others. The collaborators include Faith Ann Miracle of Microbiology. You have Chukwu Bukem John Ozo Oseido, also from Microbiology. Please, let's appreciate all of them. Understand?
The Vice Chancellor, members of the High Table, ladies and gentlemen, standing on all existing protocol, I am Madaki Enyojo Nathan, and with me here are my collaborators, Faith and Miracle and John Ozo Osiedo. We worked on the topic, Application of Vermi Technology in Solid Waste Management and Sustainable Agriculture. Introduction to Vermi Technology. Solid waste management is a general problem that needs urgent and new technology for its management. Vermi technology is the use of specific epigeic earthworms, specifically Echinacea foetida, to enhance aerobic organic waste conversion. Key products. We have our vermicast, nutrient-rich organic fertilizer containing nitrogen, phosphorus, calcium, magnesium, and diverse microbes. Worm tea, liquid biofertilizer derived by steeping vermicompost in water. And what are the benefits? Offers a sustainable, cost-effective, and eco-friendly alternative to conventional waste management method. What are research problems and significance? First, lack of efficient waste management system. It has been discovered that our conventional waste management system, which is the use of incineration and filling of landfills, has led to environmental pollution and a lot of issues. Rising use of chemical fertilizers. Chemical fertilizers has produced a lot of unhealthy crops and health issues. Environmental pollution on campus. The use of incineration has not only led to greenhouse effect and global warming, but also filling the landfills with waste had led to pollution and production of stench. And what is the significance of this research? First, provide detailed information on vermi technology application. Promote sustainable waste management. Produce agricultural byproducts for campus use. Support sustainable agricultural practices. Now, what are our study objectives? The aim of the study was to apply vermi technology for solid waste management and sustainable agriculture. What are our specific objective? First, we collected, sort, transported, and managed waste using vermi technology. We sourced for indigenous earthworms capable of vermicomposting, which is the Echinacea foetida. We constructed a mini vermi house and composter, which is located adjacent to this building. We also determined the physical, chemical, nutritional quality of vermicomposting byproducts. We isolated identified and characterized microorganisms associated with vermicomposting. And we also applied the vermicast on food crops. Now, on to our achieved objectives. First, we built a mini vermi house. As you can see, this is the go back please. This is the production process where we were constructing the vermi house. Also, we built a sustainable and suitable composter. Now, most of the composters that are known are of a non-replicable model. But this model is replicable and can be replicated by even local farmers and students. And it is very affordable. This is where the decomposition occurs. Next, we gathered and sorted waste within the school campus. As you can see, this is Adelabo Market, and the other place is our mini vermi house where we gathered the waste and sorted it. Next, we also introduced the waste and layered it. We had to layer our waste. Next, we sourced for earthworms. Like we said, we used a specific species of earthworms, which is Echinacea fetida. Next, we introduced the earthworms into the waste where the decomposition can properly begin. And this decomposition took the period of 90 days to ensure proper decomposition. And during this period, we were monitoring. This is where our supervisor was coming to guide us, and we were monitoring the decomposition. And after 90 days, we harvested our cast, dried it, and processed it, which led to the production of our vermicast. Next, this is our vermicast, the organic fertilizer that we produced. As we can see, the vermicasts are displayed on the table right there. After which, we took this, our vermicast, and then we compared it with chemical fertilizers. And as we can see, within the span of 6 weeks, not only did the vermicast grow faster and matured quickly than the chemical fertilizers within the specific time frame. We used spinach due to the short time frame on our timeline. After which, we went to the laboratory for microbial analysis of our vermicast, and we discovered a lot of organisms present. Next, so this was when we were culturing the microorganisms in our microbiology lab in University of Abuja. So we also conducted other analysis. Next, we also conducted other analysis, but before then, we did microbial analysis, and the most abundant of them was, the abundant microorganism was Staphylococcus aureus. And one of the relevance of Staphylococcus aureus in this is the fact that it shows that our vermicast is purely biological. And Staphylococcus aureus also produces extracellular enzymes like protease and lipase, which helps in the decomposition. Next, please. Next. We also did physiochemical analysis of our vermicast. Next, of our vermicast and the worm tea, which shows total nitrogen, organic carbon, and organic matter. Next, we also did FTIR analysis for functional groups present in our vermicast. It indicates complex organic composition, including carbohydrate, protein, lipids, and biomolecules. These are the results of this is the wave number and the peak number. We also did scan electron microscopy of our vermicast, which showed the structure of our vermicast, as you can see. And in this, we discovered that our vermicast contained oxygen and silicon. The presence of oxygen in the vermicast showed that this vermicast was highly oxidized, and the presence of silicon created the formation of what we call organomineral complexes, which aid the structure of the plants and the soil, which glue and helps in retaining of the minerals, the nutrients, and also water holding capacity. Compared to the organic waste, as you can see, these are the imaging we got from it. And after which, we also did the proximate analysis of the spinach that was planted by our vermicast and that of the chemical fertilizer next, sorry. And as we can see from this chart, we can see the moisture content, the ash, the crude, lipid, and everything. And from the figures, we can see that they are very similar. And why is our vermicast very important and very peculiar? With this, we can see that the same work the chemical fertilizers are doing is what our own vermicasts are capable of doing. And our own vermicasts are biological and natural, creating no side effect compared to the chemical fertilizers that are harmful to us. In conclusion, we collected, sorted, and transported the waste, and also sourced for indigenous earthworms, constructed our vermi house, tested to ascertain the chemical, physical quality of vermicomposting, and all. Recommendations. Firstly, develop standardized production protocols for vermicomposting. During the whole process, we encountered a lot of challenges. There were times we will introduce the earthworms, we'll come back and discover that our earthworms were dead. We had to discover that you don't just introduce the earthworms into the waste. There are standard procedures on how these are done. Implement regular pre-market microbial testing. Adopt advanced molecular identification techniques. Research cost-effective pathogen elimination methods. Investigate feedstock impact on microbial composition. Conduct long-term field trials in Nigerian soil. What are our key takeaways? Vermi technology offers sustainable waste management and organic fertilizer production, contingent of quality control and safety measures. And in this, I would like to acknowledge firstly God, the giver of wisdom, that helped us throughout this process. It was a rigorous process, but thank God we were able to accomplish our objectives. The Vice Chancellor, Professor Babatunde Hakim, I really want to appreciate the Vice Chancellor especially for approving the research and ensuring we were awarded the grant and to proceed. Also, the Director, Center for Undergraduate Research. I would like to appreciate firstly Professor Taibat Atoyebi, the former director that was there during the awarding of the grant and ensured we started the research, and also the present director, Professor Sunday Ejaro, who through his guidance made today a reality. I would like to thank my head of Department of Microbiology, Dr. B.O. Akombi, for his resilience and ensuring and making the laboratory available to us and all the lecturers in microbiology department. And most especially, I want to appreciate our academic mentor in the person of Dr. S.A. Aranciola. This man ensured this project came to conclusion. In fact, his resilience and discipline. There are times that I'll be weighed down, I'm tired, exhausted. He'll be like, Nathan, do you really want to continue this research? In all, this became a success. I really want to appreciate Dr. Aranciola. And to my collaborators, we fought. There were times we disagreed on different opinions, but at the end of the day, we all used to come to conclusion. To our fellow research students, our family members, distinguished audience, thank you very much and God bless you.
Well, thank you so much, Madaki, for your brilliant presentation. Please, could you cast? The Vice Chancellor would like to see that. Please, let's appreciate him once again.
I would like to welcome our mentor, Dr. S.A. Aranciola. So these are vermicast. So after the decomposition process, this was what we were able to get. So it is purely organic. The mode of application is after planting whatever you've planted, you spread it and water it. And at intervals, you continue adding it, just like the regular fertilizers. So this the vermicast. produced by earthworms.
Oh, yes. So you have a challenge now.
Cardona said, need in the society is paired with the research from the university. What he means is that what the society needs, they will now come to the university for research. Then after research of what we want, then it will now be taken to industry for production. Let me give a simple example. The farm tool we are making use of the hoe now. I urge you people to go into research. The way we are bending too much to produce, to make use of our hoe. Maybe we can invent that of India, whereby you use a long hoe, you understand, to weed. At least you have added value to the society. You understand? It's very innovative. If we can go into, the whole of this, commercialization. So it means the researcher are there, the academician, the need in the society are there, then the producer too, the industries. So you see, it's interwoven. The three, you cannot work in isolation. Thank you so much for this innovative. May God assist us. Thank you so much.
Yes, we will have to appreciate them once again, please. And I thank you so much for your understanding. But while Nathan was making his presentation, the representative of the Abuja Chamber of Commerce and Industry joined us. Ladies and gentlemen, please let's welcome Dr. Tinuke Temitope. And shortly after her, the former director of the Center for Undergraduate Research and now director Abuja Business School also joined. Let's appreciate, welcome Professor Taiba Akwe. Prof, you are being asked to gracefully walk to the high table. Thank you. Well, I've been informed that at the end of the entire presentation, we have about 6 of them here. I know that these students have been listening attentively, and you have some questions to put to them. The presenter. So we will wait patiently until then. Before I call on the next presenter, I also want to appreciate the presence of distinguished members of the press who are here to cover this event. Please let's appreciate it. Now, the second presenter this morning is from the Medical Laboratory Science. The research topic is evaluating the phytochemicals and anti-diabetic properties of curry leaves. And what I see here may be a bit shocking to me because I'm not a scientist. It's called Moraya Koyenigi. Is that what the name is? Okay, thank you so much. And is collaborating with Blackstone, Comley, also from Medical Laboratory Science. You also have Ado Mimi patients. Please let's appreciate them all. Let me now call on the lead researcher, Mr. Ibeji Paul Owa, to come forward for his presentation.
The Vice Chancellor, the Vice Chancellor, the Director, Center for Undergraduate Research, uh, the representative from NAVDAC, and the representative from Biotechnology. I want to say good morning to all of you, sir, and good morning to my fellow students. My name is my name is Egbeji Paul, and I'm from Department of Medical Laboratory Sciences. And our topic today is exploring the antidiabetic properties of Moria koenigi and the phytochemical properties. This is a financial report. Our first tranche was given to us by the Center for Undergraduate Research. That's 500,000 naira, and we use it for the utilizing and sourcing of fresh curry leaves and authentication of the leaves. And then received our second tranche, which we use for the purchase of our 20 Wister rats, streptozotocin, and other materials. Next, please. And then this is our introduction. So I know nobody's new to the word diabetes mellitus, what Hausa people refer to as Hawan Jini, and in our local dialect, everybody have a name for it. So diabetes mellitus has been an issue to the economy, and that's where our research is focusing on. So diabetes mellitus is a metabolic crisis characterized by chronic hyperglycemia, and this research evaluates the pharmacological potential of Moria koenigi, curry leaves. Locally, our mothers use this as a flavor in stews and rice and whatever, but here we are coming to check the diabetic properties of this leaf, as it has proven its worth in reducing blood sugar level. Now, the background of the study: diabetes mellitus is a chronic multifaceted metabolic disorder characterized by persistent hyperglycemia, an elevation in blood glucose level resulting from either a total deficiency of insulin secretion or a relatively deficiency due to insulin resistance. According to the International Diabetes Federation, Africa is projected to see the highest increase in diabetes prevalence globally by 2045. Now, in search for sustainable treatment, ethnobotanical research has turned to Moria koenigi, commonly known as curry leaf tree. This plant is a rich bioreservoir of carbazole alkaloids, such as mahembin, flavonoids, and phenolic compounds. So scientific interest lies in the ability of the phytochemicals to either stimulate the regeneration of pancreatic beta cells or inhibit glucose-absorbing enzymes that thereby regulate systemic blood sugar level naturally. So now this is a statement of the problem: the affordability gap, the pharmacological toxicity, and dependence on import. The affordability gap. So high-quality insulin and standard oral hypoglycemic agent are increasingly becoming an economic luxury. Majority of people we have recorded died of diabetes mellitus because they couldn't even afford the drug to cure the disease. And then the pharmacological toxicity. Sometimes people who depend on these drugs later break down with one or two deficiency, because it has an effect on them. So long-term administration of these synthetic agents is frequently associated with systemic side effects. There's always a side effect. And then the dependency on imports. The lack of standardized, locally produced alternatives leaves Nigerian population vulnerable to pharmaceutical supply chain disruption. So there's therefore a profound necessity to investigate and validate locally available, biodegradable, and non-toxic alternatives that offer a comparative therapeutic index to conventional drugs. Next, please. Now, this is the aim and objectives of this research. Primary goal of this research is to evaluate the phytochemical profile and the antidiabetic potential of methanolic extract, Moria koenigi, in reducing blood sugar or blood glucose level within a controlled experimental model. And this is a specific objective: the phytochemical characterization, perform a standardized methanolic extraction of the Moria koenigi leaves, and the experimental modeling to successfully induce a stable state of hyperglycemia in Wister rats using a single intraperitoneal injection of streptozotocin, 55 milligram per kilogram, stimulating the pathology of clinical diabetes. Then the comparative efficacy analysis to assess the glucose-lowering effect of extract over a 6-month therapeutic window and compare its potency against conventional medicine, glibenclamide. Now, the physiological assessment also to monitor changes in the body weight of the rats. Now, this is the research methodology we use for this research. So the study employs a randomized controlled preclinical experimental design, which involves a quantitative approach to assess the hypoglycemic effect of the Moria koenigi popularly known as curry leaf, by comparing its physiological response in diabetic-induced Wister rats against both positive and negative control groups. So the population of the studies, the botanical population, that was the fresh Moriah Carnegie leaves we harvested. The animal population includes 20 healthy adult male Wister rats from the species Ratus novagicus, weighing around 150 kg to 200 grams, sourced from a reputable animal house at the biological lab here in Faculty of Sciences. So on the sources of data, our primary data was gotten directly from laboratory measurement, including the percentage yield in extracting the fasting blood sugar and the random blood sugars of the rats. And the secondary data was our peer-reviewed journals. We went through a lot of journals online to compare our work and get guidelines. Next, please. Now, the methodology, now continuation. Method of data collection. We prepared our plant extract, that the leaves were shade dried and pulverized. We got the leaves after washing, we dried these leaves, and then we grinded them, and then we soaked them in 80% methanol and did our extraction. And using the rotary evaporator, we worked at the SHESCO, Shedda Science Technical College at Kwali. Now, we induced the experimental diabetic rats. So diabetes was induced in an overnight fasted rat via a single intraperitoneal injection of streptozotocin. Okay? So the dose was 55 milligrams. That's the dose of the streptozotocin. Now the treatment and monitoring, the extract was administered orally to the rats and for 14 consecutive days before the onset of diabetes. Next, please. Now this is the result presentation. This was the qualitative phytochemical analysis. So after the phytochemical analysis, we discovered that there was presence of alkaloids, flavonoids, saponins, tannins, and phenols. So these were the major, though the alkaloids were our major concentration, major focus, because they are the ones that reduce the blood sugar level. Next, please. Now, the evaluation of the streptozotocin. So that table right there was it's talking about the healthy rats. This was the mold of streptozotocin we applied, and then this was for the healthy rat, and then the test rats we applied 101 milligram per DL of the streptozotocin, and then after 3 days there was a spike in blood sugar level to 362 milligram, and that was the diabetic group. Next, please. Then this is a result presentation of comparative therapeutic efficacy over the 14 days treatment. So now, for day 0, for the diabetic control, it was 365 milligram. At day 14, for the rats, we didn't give anything. There was continuous spike of blood sugar throughout the restart. Now, for the group where we applied the standard drug, after 14 days, the standard drug was reduced the blood sugar level to 125 milligram per DL. So it was a drop by 65%. Now, for the curry leaf extract, after 14 days, there was a drastic reduction of the blood sugar level from 362 to 155 milligram per DL. It dropped by 57%. Next, please. Okay. Now, the summary of the result. The induction success was 100% of the induced diabetes rats reached the diabetic threshold of 250 milligram, and the therapeutic potency. The extract achieved a 57.2% reduction in hyperglycemia, proving that Moriah Carnegie leaf is a scientifically valid anti-diabetic agent. And the physiological impact, the extract not only lowered blood glucose level, but also mitigated the rapid weight loss associated with untreated diabetes. Next, please. Now, this is the conclusion. The research confirms that the methanolic extract, Moriah Carnegie, is a highly effective anti-diabetic agent. It significantly reduced the blood sugar level from a diabetic peak of 362 milligram per DL to near normal levels of 155 milligram per DL, effectively rivaling the performance of the conventional drug glibenclamide. Thank you for listening.
Well, thank you so much. Yes. Let's appreciate them once again. Beautiful presentation. Well, let me quickly here that the mentor, lead researcher, is Dr. Sesson Abiodun Aransiola. Please, let's appreciate it. And for this most recent presentation, we have three mentors, Dr. Obum Charity Nadi. We also have Abdul Malik Abdul Karim Omei and Professor Tony Musa. Well, thank you so much. I hope we have been listening. The Vice Chancellor has another equally important assignment to attend to, and he will be leaving us any moment. Chancellor, please let's give him a round of applause for taking time out from his very, very busy schedule to be here with us this afternoon. Thank you, Sebastian. Thank you.
I want to commend the research teams. The depth of the research, the quality, the scope is fantastic. When we take curry leaf, we like it in pepper soup. And then when someone is diabetic, say give him. Now we are seeing the objectivity. We are seeing why it is done that way with empirical reasons. Why we can extract phytochemical research, ethnopharmacological research. That's where the world is going. And we are doing it here at the University of National Unity, the Yakubu Gowon University. Congratulations, all of you. I'm very proud of the scientists. We have seen how we can develop recycled waste to develop fertilizer. This one, we are not looking at anything toxicology, but we are seeing how money. Take a problem, we are using it as for an advantage. So I congratulate. I would love to sit back and listen. I have been enjoying the presentation so much, but permit me, please. There are guests outside the university just arrived the office, so that I can. Thank you very much. God bless you.
Group photograph with the Vice Chancellor. Please kindly be on your feet so that you can show very conspicuously behind the Vice Chancellor and this team.
Sound check, 1, 2. Check, 1, 2.
Students, we appreciate you once again. And we do know that many of you have been listening very, very attentively, and you do have many questions or comments to make at the end of this presentation. As I said earlier, we have about 6 presentations, and 2 have been knocked out already. We shall be going to the 3rd, 4th, when the director is back and sitting down, other members are here. Yes, the next presentation is from, will be on the research topic, bioactive compounds, from K. Pinata and J. Tangerensis, isolation, characterization, and evaluation of anti-anemic and hepatoprotective effects in rats. And we have the collaborators in Ishaq Bashir of the Biological Science Department. We also have Edith Young Edet of the same department. And the mentor of all of these students is Dr. Palma Muta Nyadar. Distinguished ladies and gentlemen, may I have the pleasure now of inviting the lead researcher for this presentation, Ngutswen A. Emmanuel. Please let's appreciate him. I need to state this very clearly, Mr. Emmanuel. You do not have more than 10 minutes. Once it's 10 minutes, please, we will have to ask you to stop. Thank you so much. So take note of the time.
Yes. Good day, Vice Chancellor, Director, Center for Undergraduate Research, University of Abuja, invited guests, distinguished staff, and students. I am Gutu Ondunegi Emmanuel. Here with me here is my collaborator, Ms. Edidion Etim, and we research on bioactive compound from Kalanchoe pinnata and Jatropha tangerensis, isolation, characterization, and evaluation of anti-anemic and hepatoprotective effect in mice model. The following are the contents that will be examined in the course of our presentation. Next slide. Medicinal plants of different types are being identified for their therapeutic potential, and their encyclopedic knowledge is well elucidated by different authors. Some studies describe and recorded extensively on the therapeutic potential of individual plants, while others describe the enhanced biological activities arising from the first is to isolate and characterize the bioactive compound present in the equal ratio mixture of the extract. Second one is to evaluate the antianemic and hepatoprotective effects of the extract mixture. And the third one is to assess the subacute toxicity profile of the extract. Next. So our research methodology starts with the collection of plants, where we procure the fresh leaves of the KP and the J. tangerensis from local vendors in Bogolada and Boare Area Council. They were thoroughly washed under running water in order to remove the deity, and then oven dry at a temperature of 50 to 70 degrees Celsius. After then, it was pulverized and then stored in an airtight container until extraction. So, extraction, we subjected 40 grams each of the two powdered plants to 20% methanolic extraction using Soxhlet apparatus. And the resulting solution was concentrated in a water bath in order to, get the crude extract, after which were mixed in the same ratio, equal ratio for delivery. Phytochemical analysis, we run it with the help of high-performance liquid chromatography to identify about 5 bioactive compounds, which include vitamin C, vitamin B9, vitamin B12, quercetin, and caffeic acid. So we also did the hematological analysis and the histobiochemical analysis. We also did the histopathology, and then we used Excel Microsoft Word to analyze our data. Next slide. So in the experiment, we used a population of 18 healthy mice of range of weight 20 to 25 grams, which were procured from our Institute of Trypanosomiasis Research from Plateau State, and then we acclimatized them for 1 week in order to be acquainted with the laboratory condition. So after the acclimatization, the 18 mice were splitted into 3 groups of 6 mice each. And group A was the control, which received distilled water. Group B were treated with the mixture of the extract intraperitoneally, and then group C were treated with the mixture of the extract orally. And this treatment lasted for 5 consecutive weeks. So this is the result for the phytochemical analysis. You can see there is personality of vitamin C, B9, and the caffeic acid in both plants. But then you can see there's abundant in what? Jatropha tangerensis. Vitamin B12 was only detected in Kalanchoe pinnata, while quercetin was not detected in any of the plant at all. Yes? So here gives you the biomass analysis. That is the weight change during our course of treatment. We have the initial weight there and then the final weight, and that is the percentage of their weight gain. So it shows that the extract actually promotes growth. Yes? Here we have the hematological analysis. You can see that is the result. There are some superscript there, ABC, which shows the p-value using one way or another. So the difference is in the alphabet that shows that the values were significant as compared to the control group. Yes? So here is the liver to weight gain. So we have that in our control there, we have about 5.08, and the result there is tabulated. So you can see there was a slight increase in the animal treated through IP, and then a decrease in the animal treated orally as compared to the control, but that wasn't significant. Yes? So here is the histopathology. You can see the control, which is A, B is the IP group, and then C is the oral group. You can see that there is no sign of injury as per the microscopic view of the liver. Yes? Here is the biochemical, that is the biochemical analysis of the liver. So you can see the result there clearly indicate that